Okay, so hopefully you guys got up after that 10 minutes. Maybe you did a stretch, maybe you did some jumping jacks. I know you didn't, but I would highly recommend that in between videos you do some type of physical activity because sitting down is boring. So yeah. Alright, so the players in protein synthesis, because someone's got to do it, right? So the first, the most important player in protein synthesis is mRNA, or the messenger RNA. The mRNA is going to be what brings the code that's carried by the DNA. It's going to take that code, since the DNA can't leave, it's going to take that message, that's why it's called messenger RNA, and it's going to bring it to the ribosomes in the cytoplasm. So basically what happens is the DNA unwinds with the help of DNA helicase, kind of the same as replication, and the messenger RNA is created by matching up the complementary bases. So the same way that when we did replication, we paired A with T and C with G, we're going to do the same thing here. Except for some reason, and we're really not sure as scientists, but for some reason, messenger RNA, or really any RNA, it doesn't have thymine. Instead, it has uracil. So uracil is going to be a pyrimidine because thymine is also a pyrimidine. Again, we're not really sure why there's no thymine in RNA, but there's not. Everywhere that there should be a thymine, there's going to be a uracil. And so anyway, the DNA unwinds with the help of DNA helicase, and then the matching complementary nucleotides or bases are going to be added, pairing A with U, T from the DNA will still pair with A for the mRNA, C pairs with G, G pairs with C. Same thing. But also in the case of mRNA, the DNA is only going to be transcribed into mRNA either using the left strand or the right strand. So remember, DNA is a double helix. So here's my double helix. We'll say that this is the left strand and this is the right one. Okay. Technically, both of these strands are going to have the exact same message in them, right? Because they're just complementary base pairs. So they literally hold the exact same message. All of the same proteins are being coded for by the left and the right. But there's something random, and we don't know what it is, that's controlling that either the left or the right is going to be used to transcribe into mRNA, not both. And it's completely random. Sometimes it uses the left, sometimes it uses the right, but it doesn't matter because you are still getting the same message. And so the other thing that's different is that you're going to use RNA nucleotides instead of DNA nucleotides. So instead of the sugar deoxyribose, it's going to be the sugar ribose. And again, instead of thymine, it's going to be uracil. And then that messenger RNA is going to be used to find out what type of protein that we're going to make. Are we going to make hemoglobin? Are we going to make insulin? Are we going to make actin? Are we going to make myosin? All the proteins that we've been talking about this year. Are we going to make some DNA polymerase? Are we going to make some helicase? Are we going to make some topoisomerase? What are we going to make? That is going to be coded for in the messenger RNA using these codons. So codons are used to determine which amino acid is going to be put in place. Every 3 RNA nucleotides are going to code for one amino acid. Those 3 bases are called a codon. And there will be another slide in a little bit that has all of these definitions on it. Okay, so that's what the messenger RNA is doing. Now the next really important RNA is transfer RNA, and his job is also very important in protein synthesis. His job is going to be to pick up the amino acid that the mRNA was coding for and bring it to the ribosome in order to add that amino acid to our new growing protein that we're making. So it has one end called the anticodon, and the anticodon is going to match up with the mRNA. And what do I mean by match up? So in this case, the anticodon is GAA. So that must mean it's looking for the complementary message on the DNA, which would be CUU. Okay, so the anticodon is going to complementary match up with the mRNA. So for the start codon that you should be familiar with from the Amoeba Sisters that you'll have to watch, if the start codon is AUG, which we know codes for methionine, it always does, then the anticodon would be UAC, because that's what's going to complementary pair up with the mRNA codon. Okay, so on one end of the tRNA, you have your anticodon, and then on the other end is the amino acid that is going to be carried. So in this case, it's not showing us what amino acid it is, but if we went use our codon chart and did CUU, we would know which amino acid is over here on this side. All of these like loops right here, you don't need to know what those are. I didn't need to know what those were until grad school, so you certainly don't need to know what they are now. But basically they kind of just help to, this whole thing is tRNA. Okay, so one thing that we always say that RNA is single-stranded. Messenger RNA is single-stranded, not all RNA, because right here you have double-stranded tRNA. But these little loops over here basically help to like stabilize the tRNA and make sure it doesn't fall apart before it's able to drop off the amino acid. And again, I already said this, but the amino acids are going to come from our food. That's why it's important that we eat. And the tRNA's job is to essentially go around in the cell and pick up the amino acids that are needed by the mRNA message, and it's going to take it to the ribosome so that we can, again, build our proteins. Okay, so the rest of the players, they don't have very long definitions, so this is going to take up one slide. So our RNA is ribosomal RNA, and its job, along with some protein, is to make up the actual ribosome. So we've always said that like proteins do absolutely everything. They do, except make themselves. Our RNA, which is ribosomal RNA, so the thing that actually makes up the ribosome is not a protein, like everything else we've talked about. It's actually made up of RNA. And it's also not single-stranded, but different story, different day. The snRNA, small nuclear RNAs, their job is to cut out what we call introns out of the messenger RNA. Now, right now, you have absolutely no idea what oh, okay, Magnolia and Buddha. Ooh, okay, I had to pause it for a second so I could fuss these dogs fighting over nothing. So anyway, the small nuclear RNAs, again, they're going to cut the introns out of messenger RNA. I'm going to talk about introns like 7 slides from now, so just hold off, but I wanted to put it here since it's one of the players. The start codons are going to be what every protein starts with, and that would be the amino acid methionine, which is coded for by the codon AUG. Stop codons are UAA, UAG, and UGA. Honestly, I would memorize these if I was you. It would make your life easier to see where to stop a protein being built if I ask you to translate, which I'm going to do. I'm going to ask you to translate like you did in the worksheet the other day. You don't have to memorize them, but again, it will make your life a lot quicker. But if you're using the codon chart for the stop codons, it's going to tell you stop. Like, so if you look at U and then A for that first one, it's going to point to the word stop. So you'll notice stop. But if you memorize them, it might make your life just a little bit simpler. And you should definitely memorize AUG. That's the start codon, always and forever, and it's the only one that codes for methionine. Okay, so the code is every 3 bases on the DNA, the codon is every 3 bases on the mRNA, and then the anticodon is going to be the 3 bases on the tRNA. So remember, the anticodon is this guy over here, 3 bases on the tRNA. Alright, so if we were to look at a eukaryotic cell, which this whole thing is, And then right here in blue, that is the nucleus, in case you have forgotten. It's the nucleus. And inside the nucleus, you see the DNA. And then there are little segments of the DNA that are coding for specific things needed for translation into proteins that are needed for protein synthesis. So here we're coding for tRNA. Here we're coding for a protein, so that's going to become mRNA. Here we're coding for rRNA, so that's to make our ribosomes. And here we are coding for the small nuclear RNAs, which again, their job is to cut out any introns in the messenger RNA. And you do not know what an intron is yet, but hopefully I will have said it enough times that by the time I actually get to the introns, you will know what you are looking at. Okay, so again, I'm going to stop here. Seems like a good place, about 11 minutes into the video. So I'll stop here, go get some water, do some jumping jacks, pet your dog, and then meet me back over here.